guys, today we're going to be talking about DNA structure. First, we need to look at the double helix structure of the DNA. As you can see, there are 4 different nitrogenous bases: adenine, thymine, guanine, and cytosine, and those are pictured in the diagram. The 5' and the 3' ends of each structure are labeled. As you can see, there's one for each of the anti-parallel strands, and these nitrogenous bases meet in the middle and are bonded together by hydrogen bonds. And adenine and thymine are bonded with 2 hydrogen bonds, while guanine and cytosine are bonded with 3 hydrogen bonds. If you take a look at this diagram, you can see more clearly that adenine bonds with thymine and guanine bonds with cytosine. And then if you look at this diagram, you can clearly see the phosphate groups that are labeled, once again with adenine bonding with thymine and cytosine bonding with guanine. Now to move on to the actual replication of DNA. The first step is to unzip the double helix structure of the DNA molecule. This is done by an enzyme called helicase, which breaks the hydrogen bonds holding the complementary bases of DNA together. The separation of the 2 single strands of DNA creates a Y shape called the replication fork. The 2 separated strands will act as templates for making the new strands of DNA. One of the strands is oriented into the 3' to 5' direction, as you can see right here and right there. This is called the leading strand.
The other strand is called a lagging strand, and it's oriented into the 5' to 3' direction, away from the replication fork. As a result of their different orientations, the 2 strands are replicated differently.
In reference to just the leading strand, a short piece of RNA called a primer, produced by an enzyme called primase, comes along and binds to the end of the leading strand, right there. The primer acts as the starting point for DNA synthesis. DNA polymerase walks along it, adding a new complementary nucleotide basis to the strand of DNA in the 5' to 3' direction. This sort of replication is called continuous. In regards to the lagging strand, numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand, right there. Chunks of DNA called Okazaki fragments are then added to the lagging strand, also in the 5' to 3' direction. This type of replication is called discontinuous, as the Okazaki fragments will need to be joined up later. As you can see, there's a space right there where they're discontinuous. Once all of the bases are matched up, A with T, C with G, an enzyme called exonuclease strips away the primers. The gaps where the primers were are then filled by yet more complementary nucleotides. The new strand is proofread to make sure there are no mistakes in the new DNA sequence. If there were to be mistakes, that would lead to mutations. Finally, an enzyme called DNA ligase seals up the sequence of DNA into 2 continuous double strands. The result of DNA replication is 2 DNA molecules consisting of 1 new and 1 old chain of nucleotides. This is why DNA replication is described as semi-conservative. Half of the chain is part of the original DNA molecule, half is brand new. Following replication, the new DNA automatically winds up into a double helix. Just to touch up on a few more things, there's an enzyme called topoisomerase that participates in the overwinding and underwinding of DNA. With the double helix structure of DNA, things can get a little confusing, and the winding problem DNA arises due to the intertwined nature. During DNA replication and transcription, DNA becomes overwound ahead of a replication fork. Now to answer the question of where does energy come from to create the growing strand? The energy required to drive the reaction comes from cutting high-energy phosphate bonds on the nucleotide triphosphates used as the source of nucleotides needed in the reaction, and those can be shown in these 3 circles right here, or all along the DNA. As we're wrapping up, I would just like to say thanks for watching and Merry Christmas.